getting your brain poof, poof, poof. For sure you are. It's just so much information. Biology is a fascinating, subject, but it's so content driven, right? So a lot of things to keep in mind. So as I said, learn as a story, right? Create a children's book, right? With your things. So fantastic. So this is module 7 that all of you should be learning this term, right? And probably you have, this is a module that you have more fresh in your mind. So this module focuses in these skills: evaluate questions and hypotheses, evaluate investigations, conduct investigations. So maybe you were doing some practical things during your lessons, and then you had to select data from different media, and then all this is applied in the analysis of infectious diseases and what is cause that disease, how it's transmitted, how it's managed, how the organisms respond, right? Which includes our immune system. Very relevant, right? So in your questions, in your exams, you are going to have related to this topic. Probably they put questions, the inquiry questions, hypotheses related to issues with diseases. You have to evaluate investigations. They can put a table from an actual paper, and you have to analyze and evaluate that research, if it's valid, if it's reliable, if it's not biased, if it's unethical. And then you have to process some data. So that will be, for example, creating a graph, creating a table, creating a flow chart, and then all that has to be analyzed in terms of diseases, infectious diseases. So I put in there, and I got your module open in there, so you can have a screenshot of this, and your teachers can have a screenshot of this. This is relationships of all the ideas in the module, right? So all the causes of the diseases, all the treatments and control, and all the responses at immunity level. So these are 4 inquiry questions in this module: the transmission, how we respond, and plants and animals respond to the infection, our immune system, and how diseases are spread, right? And how that spread can be controlled. So verbs in this module are basically the majority investigate. So a part of other ones, but this is the main verb from this module. And in that case, when the question is going to say investigate, or the content statement says investigate, it's because for that content statement you have to plan and design an investigation, right? And you have to draw conclusions about it, right? So we are going to go through some of those. You can see that 10 out of the 15 content statements in this module are investigate, right? So in your summaries, keep this verb in mind, that you have to plan inquiry into, that is your procedures, your variables, and the answer to with a valid conclusion in those outcomes. So these are some of them, right? Transmission, investigates transmission of disease. Investigate the work of Robert Koch and Pasteur. Investigate the response of a named Australian plant to a named pathogen. So for each of these, you should have a summary focused on this verb. Make sense? A lot of work. So what I do, I usually created a scaffold for my classes with these outcomes, and each time we do a practical or we do secondary sources investigation, we answer these following these steps. So teachers, email me if you want that. Free of charge. Right. Okay. we went through this before, right? How to actually study for each module, right? Practicing from previous exams is extremely important, right? But at the same time, you have to sit down and study the actual content. And I said to you before, learn the story, right? That is my big tip. Learn the story. Over the years, and I'm teaching biology for more than 20 years, at university level and school level, learn the story of on the context. Put it in a context. It will help you to follow the ideas and have the flow chart of ideas. So if you start from the history of diseases, how all the ancient cultures were using medicine to treat pathogens, right? What pathogens they are actually, what pathogens actually they were treating, right? They didn't know anything about diseases. So how they were treating that pathogens, what type of pathogens were actually treating, how was transmitted, right? So up to here, we are in history. Then it comes the ideas of Pasteur and Koch to actually learn about the transmission, learn about those pathogens, right? What's happening with the first effects on agriculture, on society, right? So Pasteur and Koch were actually working in the industry, right? To see what's happening in there. And then from there, later on, we learned that there is an immune response in plants and there is an immune response in animals. So we are going to a cellular level. So from the history here at the top, from Asian cultures, we were actually in the macro level, right? And they were just observing infectious on the skin or on animals or plants, but they didn't understand what's going on inside that or microscopic pathogens. So, and then from there, we learn how to treat it, right? The procedures that we use for treat it first. Now we are at the level in the 20th and 21st century of control and prevention. Can you see from simple observations and maybe using Aboriginal medicine, right, at the beginning of 20,000 years ago to today is a huge change, right? And that is the impact of science in society, how we are learning over time to change ideas, right? Because that is a question that I saw in a paper related to Pasteur and Koch, how their research impacted society, right? It makes sense? I love stories, so that's what I created this. So those, if you were learning the story, right, you can answer the inquiry questions in each module, and we are going to go through each of those. So the first ones are, how are diseases transmitted? And the first things in there is you have to learn how the classification of pathogens, right? The textbooks and Google images have beautiful, excellent pictures of these and excellent tables. The classification of pathogens, the characteristics, and the transmission adaptations. So we'll focus in transmission adaptations because you have to learn how that pathogens invade the other organism, right? What are the things that that bacteria, that virus use, what strategies they are using to get inside the cell, to get inside the organism, how it tricks the organism, right? That by the time that we feel sick, that bacteria, that virus is inside the organisms and make it sick, right? So do they have inhibitors of the cell membrane enzymes? Do they actually compete with other protein channels in the cell membrane? They mimic other substances in the body of the organism, inhibit enzymes, right? So in this case, you have to do a little bit of research, right? If the textbooks are not explaining this properly, do a little bit of research. What are those adaptations in those pathogens? Learn adaptations for different types of bacteria and different types of viruses as well in plants and in animals. Then for the work of Pasteur and Koch, I just forgot what I was doing if I changed it in different way as I did it last year. So from Pasteur and Koch, you need to actually, probably you're going to do the experiments in your class, the Pasteur experiment. It is an actually outcome from Pasteur. Please write the experiment with the aim, the inquiry question related to it, variables, the procedure, right? Learn how to outline a procedure. In the HSC, you will have questions that outline a procedure, or in class, you perform an experiment based on Pasteur research. Outline a procedure. You don't need to put all the number 1, 2, 3. When it's outlined, you can just write in sentence that makes sense, and it has an order, a chronological order of the steps, right? In that outline of that procedure, you have to put the dependent, the independent variable, the control variables, right? All the variables in there, and how was the conclusion? What's happening at the end? How is the conclusion of the practical for both of them? With the Koch work, you need to know the postulates. You need to know the order of those postulates, right? And how those postulates relate or can be used in today's things. What is the impact of these two in today's society? Because we're still using these procedures today, right? After 200 years, right? So learn that. What is the impact? Improve the quality of life of people, because we understand how pathogens work. We understand how to identify a disease using the Koch postulates, right? So those ones are the actual impact that they have in the society. And then the next one is causes and effects on transmission, all right? So learn some epidemics and agriculture cases. Please learn about COVID, right? Please learn about the Spanish flu. Please learn about other epidemics and pandemics that we have in previous years, because we can compare the impact and how those pandemics were for longer due to the lack of technology, and how this pandemic is sort of controlled, right? In a shorter period of time, right? So the World Health Organization web pages are really, I recommend it as a resource to analyze these cause and effects on transmissions. Okay, the other question that we have in this module is, how does a plant or animal respond to infection? So we need to learn one example well related to fungal or viral pathogens. There are not much information in this, because actually Australian plants are very resistant naturally to viral pathogens. They are not much to fungal, but yes to viral pathogens. So you're not going to see many research on this. There are some examples in the textbooks, but there are not too many examples. But you need to learn, usually focus on the fungal and have some idea of the viral pathogens as well. So how the plants responded is related to if they create a cyst around, similar to the cyst that they create in humans. I can talk, a lot about plants because it's my specialty. But anyway, so they can create a cyst. They can drop the leaves of the disease section. They can isolate and cut off the xylem and phloem flow to that area, so that area will die, right? That is an advantage comparing with an animal, for example, that is very rare that an animal is going to cut off the flow of blood to a limb to lose it. It can happen, all right, but it's not a common strategy for the animals. So the immune system of plants is more a responsive, reactive response to the fungus or the virus, comparing with animals that we have 3 lines of defense, right? All the animals have 3 lines of defense: the physical barriers and the chemical changes, right? When we have inflammation, fever, excess of temperature, to kill that foreign object in our systems. Again, when you finish learning about this, how does a plant or animal respond to infection? These are the evidence that you have to put in your answer. And then we focus in the human immune system response, right? So we have the innate and the acquired immunity and the primary exposure to a pathogen. Differentiate clearly between innate and acquired immunity, because with COVID, there are going to be questions, right? Differentiate well all the steps, right? From the moment that a pathogen gets into our 3rd line of defense, what are the relationships between the T helper cells, the B cells, what are the characteristics of the B cells, the T cells? That is going, if you have a question in the HSC, and if the question said describe the human immune system response to a pathogen, right? They can be that inquiry question. The differences between a BEN 4 and BEN 6 will be if you just name the T cells, that will be a BEN 4. But if you name the T cells, the B cells, and the relationship with them and how they interact and what are they and what they do, that is a BEN 6 answer, right? So keep in mind all the steps in the immune system, right? Do flow charts, do graphics, do infographics, right? There are plenty of things in the internet that you can use to study this. Right, and then the last section is related to prevention, treatment, and control. What are the spread factors, right? Is related to physical contact between individuals, is airborne, is is just because a person sneezes on another person. It just lists all the spread factors that they are related to bacteria and viruses and fungus, right? What prevention strategies do they have, right? Do we go to quarantine? Do they take antibiotics? They take antivirals? They just use topical things on the wound to prevent bacteria to grow. Just use and list and learn specific prevention strategies for each of the type of pathogens. Then with the antivirals and antibiotics, just focus in the use. You need the definition what is an antiviral. You need a definition what is an antibiotic. It was a couple of years ago a question related to what to use. They described an infection by a bacteria. But if you didn't read the question properly, you can put either of those two. But in the stimulus, it has the picture of a bacteria, not of a virus, right? So you have to use an antibiotic and how that antibiotic is going to work. So the management of the spread related to the environment, right? The isolation of the area that is infected, the isolation of individuals, the quarantine effect of those individuals, right? So they can be related to the flu that we have, the bird flu, the current COVID situation. So just keep an eye on that. Incidence and prevalence, if the teachers have access to previous textbooks, there is a very good examples and very good explanation of the protocols to follow in epidemiological studies, right? So if in the previous textbooks there are very good examples of this and how to manage this. So the incidence and prevalence in this case focus malaria, dengue, and the mobility and immunize of humans, right? So they can ask why Australia, is closing borders. That is to prevent the mobility, right, of humans. The immunization procedures that we have, for example, related to malaria, related to dengue, and of course related to pandemics as well. Learn the terminology, what is mortality, morbidity, incidence and prevalences of diseases. Create your own definitions for that. And then it says history and cultural strategies to control the spread of disease. As I said before, learn previous epidemics, learn previous pandemics, learn what's happening, during the First World War when people have a wound and they can just put vinegar on top, comparing to later on, Vietnam War and Afghanistan wars, that we have so much technology to treat wounded soldiers, right? So if you want to use the wars to see the history of the strategies to control infections, and then use the Black Plague, the Spanish flu, and COVID to see the spread of the infectious diseases at global scale. Aboriginal protocols to develop medicines and biological materials. The Canberra Institute of Aboriginal Studies, they have very good resources for this, and I think I put it at the end of this presentation, the link to that place. All the protocols that you have, for example, to grab lemon myrtle from the bush, or eucalyptus leaves. There are industries all well established today related to that, but it can happen that maybe a new pharmaceutical company discovered that Aboriginals in the north of New South Wales are using different type of plant as an antiseptic, and they want to exploit that active ingredient. So what are the protocols? What are the consultations that those Aboriginals has to have and go through, and the acceptance of those pharmaceutical companies first to collect from country and place, and then to reproduce that active ingredient from that plant and create a pharmaceutical from it. 90% or 95% of all the medicines that we are using today in the 21st century are coming from plants, including chemotherapy, right? So one of the active ingredients in chemotherapy for lymphoma is actually the mustard seed, right? A very toxic mustard seed, and 2 mushrooms that they are very toxic to kill cells, right? So pharmaceutical companies extract the active ingredients from plants and fungus to create drugs to treat diseases, infectious or non-infectious, all right? But there are a lot of protocols for that, right? Elders has to be consulted. The entire Aboriginal Council has to be consulted. So there are a lot of things that you have to follow. So please follow the link that that I put in at the end of the presentation. Okay, how are we going? Are we traveling okay? Too much information, right? Cool. So, exam type questions related to this topic. I divided the questions into content questions, skills questions and integrating modules questions, right? So let's see. These are going to be content questions. These are coming from paper not last year, the year before, right? So answers. Which of the following is an example of a non-infectious disease? And it should be infectious disease. Okay, perfect. D. And this is related to malaria, right? It's a disease in humans caused by a single cell Plasmodium species. Which of the following is true for malaria? I think you got the answers in the booklet, right? You're cheating. Right. Keep in mind, right, and read the question question properly. It is transmitted by female. Which of the following is true for malaria? Right? So the mosquito is a vector, the Plasmodium is a pathogen, and then the mosquito is a pathogen, and the Plasmodium is a vector. So as Tim before said, there are going to be distractors in the multiple choice questions. One, it will be totally wrong, but the other three, if you don't read it properly, could be a little bit tricky, right? So take your time. You don't know the answer, or you are not remember the answer, or you are not sure about the answer. Mark it on the paper and then continue, and then you will come back later on, right, to that. And maybe as you go through the paper, your brain is going to get organized with the information. Say, oh yes, now I know exactly what is happening, right? So don't waste and get stuck in one multiple choice, right? You have only very short period of time to do it, right? So just go through slowly. This is from paper 2018, right? So causing disease has been unicellular, having cell wall, no nucleus. This relates actually to what you learn about unicellular organisms in year 11, right? Well, we got A. Beautiful. Excellent. Now we're going into questions that are related to extended responses, right? Second section of the paper. This was paper 2019, and it says outline one adaptation of the specific pathogen that facilitates its entry into a host. It says outline, so you just have to give the characteristic. No explanation, no analysis, very simple, one adaptation, right? So you have to name a specific pathogen, right? And name the specific adaptation. You can see the marking criteria in there. One adaptation and then name the specific pathogen. But if you just name a specific pathogen, you will get 1 mark. Or if you just name an adaptation, you will get 1 mark. That's what we call in HSC marking provided some relevant information, right? So that is an example from the paper. So they use a bacteria that it grows in the stomach of, actually it was positive for this bacteria. I had to take antibiotics for a long period of time, right? And that is the characteristic of the pathogen and what's happening and how it helps to penetrate the bacteria. You can see that the answer was 2 sentences, identify the bacteria, right? And gives the characteristics of that bacteria. Have a go, right? Eventually, when you are practicing things, you can use other examples that you are not going to read, that is not exactly this one, right? Now, as I said before, learn 1 or 2 examples well. You will need them for the HSC. This is for the paper from last year. It's related to cholera, outline 3 strategies that could prevent the spread of cholera. It says again, outline. Very simple strategy and a very simple description of the strategy. Nothing too much in there. However, it is better for you to reach that band 6 to put the next verb in here, which is explain. Right? This is a tip from under the table, right? A little bit of a corruption. So, to get that band 6, if you see a verb that is low-level verb, like this one, answer your question as it was a higher order verb, right? As an explain verb. That is going to help you to get that band 6 because you're going to put more information, right? So, 3 strategies. Washing hands, purification of drinking water, and proper disposal of sewage. However, if you listed the 3, right, you're going to just have 2 marks, right? If you actually list 2 with the how you spread it, you get 2 marks. But the key point in here is this second section could help prevent the spread of cholera. If you just listed and you didn't put the how prevent the spread, you just get 1 mark. Become familiar with what is expected in the answers, right? Use the HSC biology packages when you are going to practice from previous exams questions, because this is going to become automatic in your brain, saying, oh, it is an outline, it has 3 marks, right? So if I explain each of the strategies, I will get full marks, right? It makes sense how it works in the marking criteria for us. All right, the next one are exam type questions with skills, right? You are going to identify these questions because they are not focused in the content per se. They are focused in what actually you can understand from investigations, how to read a table, how to make a graph. Right? So, first one. This is related to islands that they are separated, you know, in different positions in the sea. They have different policies of quarantine, and then a nearby country, this is very relevant, right? Has an infectious disease in its stock. They created an investigation to see which of the policies is going to be most effective, right? And then which one is going to be a suitable design feature of the investigation. Which one? The control is the smaller island, the control is the number of infected cattle, the independent variable is a quarantine policy, the independent variable is a number of infected cattle. Answers? A, B, C, or D. Right. Excellent. Can you see how can you get a little bit confused with the 4 answers in there, right? So if you don't have very clear what is a design of an investigation and what is needed in the design of investigation, right? You can actually stuff it up in here. Right, another one. Again of skills, right? Students design, this is from the paper last year as well. Student designer conduct an investigation to test for the presence of microbes in 3 different food samples. This is an experiment that you actually did in your schools, right? Testing for microbes in foods. They inoculated agar plates with the samples and you incubated. Which row of the table represents a valid design, again on design, for the investigation? Well done. A, food sample, independent variable. We are testing the number of microbes and the control is the agar plate with our sample. This is a tip for all of you teachers. Now at university level, they are doing something. Instead of the students actually marking the correct answer, they have to justify every answer. So if that is the correct answer, why? If B is incorrect answer, why? And so on. So I was tutoring a girl the other day she's from the University of Melbourne and she's studying nutrition to become a nutritionist, right? And her assessment was all multiple choice questions that she has to justify every single answer if it's correct or incorrect. That could be a very good practice, right? So it was a very interesting assessment task that I was actually helping her to answer all the justifications. All right, this is coming from the paper 2018 and it's designed on the new flu vaccine, right? And the efficacy of that flu vaccine over the years, right? Draw an appropriate graph to represent the data. You cannot imagine how many line graphs we have in here. Why it cannot be a line graph? Be brave. Why this type of data, it cannot be a line graph? Chunks of data. Very scientific, Miss Park. Yeah, yes, chunks of data. They don't have a direct relationship over time. That's a good point as well. The actual data is discrete, is not continuous. That is a key point when you're going to observe data and decide if it's a line graph or a column graph, right? Because there are non-infinite numbers between the first value and the second value. It's actually discrete, it's actually fixed. When you are measuring temperature over time, the temperature could be 20.3, 20.35, 22.34, right? So there are infinite numbers between one value and the next. But in discrete numbers, in discrete tables, data tables, the numbers are fixed and they are usually, as Ms. Sparrow said, they are in chunks, right? So you can see there is a range in here between 1 year to the next, right? If we are measuring temperature over time, right, in this case, we can create a line graph. Make sense, Mike? Keep that in mind. It's extremely important. That is the application of math in science. Right. So this is the actual answer. It's 3 marks. They are going to 1 mark for draw a column graph. If we saw a line graph, this question will be actually 0. Right? Because it's a skill question. They're asking for the axles to be labeled properly. Please don't forget the units. That is a silly mistake that a lot of you have in your exams, right? And please add the title. because sometimes adding the title doesn't hurt, right? And because you are not going to see the marking criteria, just sometimes for the for NESA to be picky, they want a title, right? So add a title just in case. And when you're going to draw the graph, try to be nice and neat. Keep an eye on the scales, right? As well, that they are evenly distributed from, every 10 in this case. Label your graph properly. And if you have to annotate it, for example, that in 2010 and 2011 there is a peak on the effectiveness of the vaccine, maybe you need to circle the top of that column and make a comment in here related to it, if the question is asking you. Right. For 2 marks is because some of the axes or some of the scales or some of the units are not present, right? So you're going to have marks down for that. I know, Ms. Vargo, that is really interested in this case, but I saw marking criteria in previous exams that they were actually putting marks for the title because they want to know if the students can synthesize, right, the question into just a simple title, right, and relate variables in the title. So very interesting. They can be really picky if they want to, right? And we as a markers, as the team said, when we get the marking criteria, that is set on stone. We cannot argue about it, right? Already NESA discussed that with the people who wrote the exam, people who are the assessors of the exam. So when we get it, we are the slaves, we just mark, right? Cool. This is another skill one. This is coming from last paper. It is identify 2 features of the method used that contribute to the validity of this study. So the study is related to the exposure to arsenic, right, in drinking water. Over time, they created an epidemiological study, right, and it gives you the sample in there and what they did, what they were testing, and then the graph represents the survival rates for males and females over 11-year periods. So identify 2 features of the method is one question, and the second question says discuss the data presented in the graphs in relation to the hypothesis. The hypothesis was that the arsenic in the drinking water actually increased the mortality of people. So these were the marking criteria for the 2 features of the validity of the study. Large sample size is the first one that you should put in there, right? And then the arsenic exposure, the collection of the data, right? And the different parameters that they were collecting, the age, the sex of the people, right? And the socioeconomic status, okay? So just identify, you have to name it. You don't have to explain anything, right? Then the second question, which is 4 marks, and in the questions, just you need to provide points for and against the hypothesis and relate points made to the data, right? So what it means provide points for and against of the hypothesis. Do you support the hypothesis? Why? You don't support the hypothesis. Why? Right? And then you have to relate that to the data. Means that you have to refer to the graph. Right? I think you got this in your book, so please, this is very small. But if you read the sample answer, right, we can actually identify in here those requirements for the band 6 descriptor, right? So the first sentence is describing the results, so a little bit. That is giving you the background information to support or not support the hypothesis, right? Remember that one of the descriptors was to analyze data, okay? So in this case, we are analyzing those graphs, right? And then what's happening with the gender? And then what's happening with the doses, right? All related to the gender. And then other ones are and he puts another unfamiliar situation in here is the nutritional state of those people or the genes, the genetic makeup of those people, right? And then survival decline progressively over the 11 years, which supports the idea that the arsenic exposure increases over the years. Survival declines, right? And then again a little bit of the results. So you can see in here the amount. This is a simple answer. Maybe you can have a different one, that is okay, right? But for those 4 marks, the actual marker will look for all these characteristics. So points in, some people were saying no, you know, if you want to reject the hypothesis, the answer has to have all the evidence why you are rejecting the hypothesis. We are not penalized that you are going for the negative of the answer. The answer can have a positive or a negative answer, right? So we do not penalize for that, but if you go for the negative, all the aspects should be there, right? And constantly refers to the data. This is another one that is coming from, and I put it in there because it's focused on models, right? This is actually a model of a way to modeling the disease, the transmission of a disease, right? And then it's referring to a multiple choice questions, right? And then the next one, oh, I thought I took it out. You can have the same diagram referring to an extended response, right? So they can use it for 2 things, right? The same diagram in a multiple choice and then the same diagram later on in a extended response. So keep that in mind. Another one that is extremely important that I went through at the beginning of the day was the annotated diagrams and graphs. This is actually coming from the paper in 2019, and you can see that the graph was just draw, then you have to continue drawing the graph and annotate it. It is a very tricky question, right? So they said continue the graph, right, to show the immune response upon I cannot read. Oh, upon new exposure to the same pathogen, right? So what's happening? And you can see primary exposure, secondary exposure to the pathogen. So in here, you have to have a very good understanding of the immune response. And you can see in the model answer how the students, well, the teachers who wrote this, make inferences of how the memory T cells work and how all this is happening, right? You will have plenty of room in your exams to draw things and annotate things. And it says use annotations in the diagram. So you have 2 marks for the line going up and down, so this section, and then 4 marks for all the annotations. So the marker will look for actually what's happening at the moment of exposure, the first exposure, and then each step of the reaction of the immune system in the graph. It is a nasty question, right? Yes, Gail. Yes, it is okay, Gail, as soon, but just make sure that the line is very clear and dark, because your paper will be scanned and not all the photocopiers and scanners in the schools have the same quality and the same resolution. So just make sure that your line is very dark for it. The same when all of you draw diagrams and things like that. Now, the other thing that it was in that Band 6 descriptor was the unfamiliar situations and interrelatedness between concepts and context, right? So this one, for example, and it was on paper 2018, is related to cancer, right? So it's explaining cancer in here. About melanoma and how they are treating, and they created vaccines with antigens against the cancer and things like that. So, and this is a multiple choice question. Multiple choice questions as well are divided according to the BAMS descriptors, right? And the ones that they relate to stimulus usually are tackling that Band 6. You don't necessarily need a question that you have to write a mini essay to have a Band 6 answer, right? It's all related to tackle those requirements in the Band descriptor. So, what can be inferred from the scientists discover? And the effect of the melanoma vaccine is to stimulate what? So you can see that these they are relating infectious response of the immune system with a vaccine, right? They related to non-infectious disease, which is cancer, right? And they related to skills. Infer what you can do from the science discovery, right? So, cancer cells carry unique antigens are the first one, and the second one is cell division to produce more lymphocytes. You need to read, this is a tricky question, you need to read, particularly in the answers for question 20, and these are the last two multiple choices in the paper. Read this. If you're not sure, just mark it as I said, and then come back later when your brain starts getting more organized. All right, this was last paper, and this is so unfamiliar to all of you because we do not teach this, right? In a way, we teach different types of, we teach the basics and the protein synthesis, right? We teach you what is the structure of a virus, of a pathogen, right? An RNA virus, right? And then in here, do not whinge, as Tim said to your teachers, oh, my teacher didn't teach, how do you know viral reproduce, blah, blah. We don't have to, right? These are tackling that unfamiliar situation again, and is relating to different modules. So the diagrams shows you the structure of the virus, and the diagrams shows how the virus uses the protein synthesis in the host to reproduce itself. Cool. The question says, so it said after infection, so it gives you more information in here, and then it says use the information provided in diagrams 1 and 2, explain the role, listen to this, of viral RNA polymerase in the reproduction of the virus. Viral RNA polymerase is an enzyme similar to the ones of the protein synthesis, right? When the DNA is translated and transcribed. So, can you see that if you don't know what is an RNA polymerase or DNA polymerase, you're going to be in trouble, right? And this is coming from the first module, right? At the back of the exam packages, you have the list of the questions that they have actually, and it says in there what modules are actually tackling that question. So if you want to know question specifics to relating different modules, you go back to the guidelines in the package, in the exam package, and you will have the list and the grid for what questions are tackling each module, right? So the marking criteria in this was 5 markers, and it says explain the role of the polymerase in reproduction of the rabies virus, reference to transcription, right? Production of proteins and viral RNA replication. So you have to interpolate what you learn in protein synthesis to a viral reproduction. That is the interrelatedness, right, of the thing. So you need to think outside the square when you are learning these things. And please, teachers, expose your students. I know that it's time constrained, but try to have tutorials at lunch and sit down and analyze this type of question. I don't know. You use the strategy that it can help the students to have that thinking outside the square, that critical thinking, to apply the knowledge that they know into a new unfamiliar situations. And then relates newly produced proteins to RNA polymerase and viral RNA replication, right? So in this case, with the actual answer, the first sentence is explain what is the actual function of the viral RNA polymerase, right? What is happening? So what is the enzyme? What is happening with that enzyme, right? And then what is actually the entire procedure, right? So you can see that the answer is not very long, right? But it has to be clear and succinct, right? And you can see the links. This complementary strand is then used. That gives the marker the link from one idea to the next. Which is another thing that it links things, right? So learn some of those links. Have those links under your sleeve, right? When you are going to actually answer the questions. So summarizing, 2 minutes, I think. Did I finish? 120? Yay! Right. Summary for this module. Answer the inquiry questions with all the evidence below those inquiry questions. That will help you to interrelate all the content. Learn the verbs. Keep an eye on the verbs that they are in those content statements in your syllabus. Integrate things. Sit down and say, okay, I learned about diseases. This can be related to this module because that module because that. Put examples, all right? I know that is a lot of work, but you are in the HSC level and the marker will expect that you did that when you are studying. Skills. Learn about the investigation process, right? How is the scientific process? How to read and make graph and tables, right? The verb for tables is tabulate, right? Annotations of models and graphs. And then practice from previous papers, guys. Practice, because if not, you are not going to become familiar with the wording of the exams. Oh, thank you, Miss. Thank you. With the words of the paper and what is expected in the paper, right? If you're sitting in front of the exam and it's the first time that you see the paper, you're going to panic, right? All right, any questions, guys? We have 0 minutes to go to. Hopefully this is helping all of you to clarify, all right? Tim has a different approach of what I used in my presentation, and then the next teacher will.
Great. Thank you so much. Thank you. I think everyone's probably feeling a little bit
Have a different
brain
approach
fried
that is giving you
because there's a
different
lot of
strategies,
information
right? A
there.
different
I know
idea
I'm feeling a
on
little
how
bit brain
to
fried.
get
So
all that
we will
to learn
take a
and to study.
break until
Right.
we're coming back at 2 o'clock, guys. You've got a 40-minute lunch break. So go and clear the brain and
Oh,
have a chat. And
it is. Right.
thank you. And these presentations will be available to your teachers
Fantastic.
through Google
Don't mind
Drive link that's going to be shared at the end of this
emailing
presentation.
them
Thanks,
if
everyone.
they need